start off by saying a really big thank you to the UK Plant Sciences Presents teams for organizing this great way to take advantage of the new normal and bring together plant scientists. I'm excited to tell you a bit about what I've been doing for the last couple of years, and what I'll cover today is really the work that I've been doing to bring agricultural weeds into the molecular lab. But to give you a bit of context, I wanted to start off by explaining to you why I think this is important and why I think it's a good thing to be doing. So in evolutionary biology, there is a delay. There we go. In evolutionary biology, we have the Red Queen hypothesis. And this theory is named after a character in Lewis Carroll's book, Alice in Wonderland, who, among other reasons, is famous for the quote, "Now here you see, it takes all the running you can do just to keep in the same place." This theory explains the idea that in order to survive, a species must run as fast as it can to be constantly adapting and evolving, because other species it's competing against are also adapting and evolving as fast as they can. Therefore, if anyone were to stop, they would be left behind. So all that running is required just to keep in the same place in the food chain. It's most often used to explain things like host-parasite relationships or predator-prey interactions, but I'd like to argue that agriculture is a very good example as well. For really, there is a lot of running required to make and maintain our agricultural monocultures. So a monoculture like this is really a challenging thing to maintain, because if we were to stop running or even slow down just a tiny bit, we end up with a field like the bottom image. So the only difference between the desirable monoculture on the top and the messy field on the bottom is that the bottom picture was taken in a section of Rothamsted's long-term experiment where we do not use any herbicides. All of the rest of the inputs are the same, just no herbicides in this section. So this one management change, a tiny slowing of the pace, has allowed for the competing organisms, the weeds, to catch up with the crop. Now, you could argue that this isn't a fair comparison, because this section of the field has never received herbicides in its 177 years of being a field, and therefore this situation isn't something that will happen in our fields in a relevant time frame. But plants are pretty good at capitalizing on mistakes and opportunities. To illustrate my point, compared to the long-term experiment, these two new images show good examples of what's called a spray miss. Now, a spray miss occurs what happens when human or machine error means the herbicide hasn't been applied correctly, and it's often just one pass of the herbicide. Now, I'm showing you this because these images illustrate just how quickly and efficiently these yellow flowered weeds have capitalized on these mistakes and caught up with the crops. So it really doesn't take much for the monoculture to fall apart. But when weeds in a field cannot always be blamed on things like machine error or human error. Sometimes the weeds catch up even when we do absolutely everything right and everything possible to maintain the monoculture. And that's precisely what's happened here. So herbicides have been used on this wheat field, but this weed, blackgrass, has evolved resistance to them. In fact, blackgrass can survive up to 7 different modes of action. Now, I'm not a farmer, I'm a molecular geneticist, and this means, although I'll probably get in trouble for saying this, I have admiration for these particular weeds. The thing I like about them is the traits that makes weeds weedy also makes them particularly interesting. I'm awed by the fact that in about just 30 years or so, blackgrass has evolved the means to survive and even thrive in our agroecosystem, despite the fact that we're constantly trying to kill it. So despite the odds, it has found a way to survive. So it's my understanding that I believe that by understanding the genetics of weediness and getting a mechanistic understanding of what allows them to survive despite those odds, we can identify exploitable strengths and weaknesses that can be used to create novel weed control strategies and to inform crop breeding programs. Both approaches are working towards sustainably enhancing agricultural production. So here's my take on that famous illustration where the Red Queen is agriculture and pulling Alice, our poor wheat, along with her. Because they continue to run, they can keep ahead of the weeds, but if they stop or slow down, or if the weeds find a way to get faster, then the monoculture falls apart. So now the most defining feature of weeds is that they're particularly resilient, and they often find a way to run faster. And blackgrass has more than demonstrated its prowess. In the UK, we have a small handful of problematic weeds, but really blackgrass stands out from the rest of them because it causes serious problems for farmers. Across the UK and Western Europe, it infects winter cereal farms like wheat and barley. And herbicide-resistant Alloprecursus myricetes is present from Spain to Sweden. Although in many of these countries that's actually still a developing problem, where it is present, it causes significant reductions in yield and increased costs. Herbicide-resistant blackgrass is a very costly problem, and a recent report found that farmers lose nearly £500 million of income to it annually, and it reduces wheat harvest by 820,000 tons, equating to 1.2 billion loaves of bread. So eradicating blackgrass would be a major step towards closing the yield gap and reducing agricultural inefficiencies. Although the impact of this weed is high, it's also just a very interesting plant to study. I've told you that it has widespread resistance to multiple herbicides and that the grass weed grows in grass crops, so it's able to circumvent both chemical and physical control methods. But it's also a really competitive weed with a life cycle that completes within the winter annual harvest. This means that it outcompetes even our most elite cultivars, and the seed bank is replenished annually despite our cultural controls. So none of the weed control measures that we currently have in place independently are effective. So this seems like sort of an intractable problem, perhaps, and so you probably are wondering what I'm going to do to get on top of blackgrass. Well, my plan is to come at it from a totally different angle. I want to bring blackgrass into the lab and gain a molecular level understanding of its weediness in the hopes that we can find exploitable strengths and weaknesses. My approach is driven by the fact that pre-blackgrass, my research questions were focused around understanding how plants survive their environmental challenges. I used to work with Arabidopsis, and I was uncovering the molecular responses that allowed environmental inputs to become developmental outputs. My plan is to be able to do everything that I used to be able to do easily, or relatively easily, in Arabidopsis and agricultural weeds, because really these weeds are just doing very successfully what I used to study in Arabidopsis, but they're plants that cause real world pain. So blackgrass is not a model organism, but it's a pretty good system to work with. For instance, as blackgrass grows wild, we have great resources for natural variation studies. It's experimentally tractable, it has high fecundity, and as I've explained, real life relevance. However, when I started working on blackgrass, what became very clear very quickly was that we were lacking things to do genetics and genomics tools. So this is where I've focused my efforts over the last couple of years and where I'll focus for the rest of the talk. One of the things I'm doing to address the lack of genomics tools is as part of the International Weed Genomic consortium on the executive committee. This is a group of like-minded academics and industrial scientists who all have interests in developing the genomics of weedy species. This group has obtained funding to do public-private research aimed at closing this knowledge gap and developing the critical resources that are required for major weeds through partnerships and training. We not only recognize that our knowledge of weed genomes lags far behind crops and other plant species, but that this lack has a significant hindrance to progress, as weeds have a unique complement of traits that distinguish them. We recognize that there's unique questions in weeds that cannot be addressed using model, wild, or crop plant species. For instance, the evolutionary processes that govern resistance, gene flow, seed bank dynamics, and stress tolerance are different in weeds because of the unique selection pressures that they face within the agro-environment. Therefore, the IWGC aims to enable science to be done in weeds by developing the genetic and genomic resources and a trained workforce to analyze and implement innovation in the field of weed science. To fulfill our main purposes, we're generating the tools necessary for weed genomics and have three main objectives: to obtain reference genomes for the most important weed species worldwide, to provide user-friendly genome analytical tools and training through web-based data and resources, and to facilitate discussion and collaborations within this emerging field. Within each of these activities, we aim to ensure that we inspire new scientists to come and work on weed genomics. So if you're feeling inspired, do get in touch. But back to blackgrass specifically, and using it to illustrate how far behind weed genomics are. So about a year ago, this image from 1944 was the best information that we had about blackgrass genome. Since then, we formed a collaboration with Bayer Crop Science and Clemson University to sequence and annotate the blackgrass genome. This is a large diploid genome, and it's about 3,500 megabytes. By Illumina short reads, we now have almost total coverage and assembly sizes that match our flow cytometry. Fortunately, the number of pseudomolecules that we have generated also match the observed number of chromosomes, and their sizes do match. The BUSCO scores for both the genome and the proteome are really high, and the repeat densities and number of predicted coding genes are in line with what's expected. This all gives us a very high LAI score, and altogether, our genome is at the gold standard. We're very proud of this and hope that it will be published relatively soon. But of course, a genome is more than just a sum of its parts, as it enables us to answer interesting biological questions. For instance, I mentioned that we have very good resources for natural variation studies. At Rothamsted Research, we have a very large collection of blackgrass germplasm that have been collected annually since 2014 from across England. And for these, we've accumulated management, genotypic, and phenotypic data. For instance, we've determined the number of herbicides that each population is resistant to. I've taken some of these populations and done full transcriptomes for them. Now, the aim here is to identify genes that are differentially expressed between populations with different phenotypes, but similar within ones with similar phenotypes. With this, these experiments can get to the root of why one population or set of populations exhibits a different set of phenotypes than others using unbiased and phenotype-driven methods. So creating and analyzing these multifactorial combinations of quantitative, genotypic, and phenotypic data is not so straightforward, and we're working towards gaining some good funding to develop these new computational bioinformatic approaches. So this is one project where a genome will enable hypothesis formation, because rather than just dots on a volcano plot, we can actually figure out what those genes are and what they do. So the genome and the transcriptomes that I just talked about are building access to omics. But the other thing that we really needed in weed science was a means to do reverse genetics. We needed something that was quick, something that was high throughput, and that would alter expression throughout the entire plant. Among the myriad of techniques that I could have gone for, I chose virus-mediated reverse genetics. So these are dominant and transient techniques that have been around since the early 90s, and they're tools that rely on plant-virus interactions to transiently alter gene expression. In short, they use a genetically modified virus that is used to either prevent protein production by degrading the plant's messenger RNA through RNA silencing pathway, or to introduce an RNA sequence into the plant so the protein is made. These two approaches are called virus-induced gene silencing, VIGS, and virus-mediated overexpression, VOX, respectively. Both cause systemic infections with the GM virus, and they result in expression changes that are mostly manifest in the new leaves. Now, I wouldn't be bothering to introduce this to you if it hadn't worked. So here you see VOX in action in blackgrass. The plants on the left were infected with a VOX virus that has just the multiple cloning site, and with a UV light and filter, you can only see the autofluorescence. Whereas the ones on the right were infected with the VOX virus carrying the GFP, and you can see the characteristic GFP fluorescence. Now, the one thing to point out is that not every cell within every leaf or every leaf is glowing. And this is a limitation of these virus-mediated techniques, as the phenotype is only manifest if the virus can replicate within the cell and drive translation of the insert. Therefore, although it's patchy, this image demonstrates that VOX does work in blackgrass. Of course, I have all the molecular backing to show that this is the right thing and it's virus-mediated heterologous expression of GFP. However, I'm not going to show you those data here. Now, glowing blackgrass is really interesting, but it doesn't help us to understand which genes underpin weediness. For that, I decided to borrow something from my many years of creating transgenic Arabidopsis. For many of you, Bosta or glufosinate is the one herbicide that you will know, and this is because Bosta the resistance gene is often used for selecting transgenic Arabidopsis. In this case, I used VOX to drive the bar gene, and the VOX infection was sufficient to confer herbicide resistance to blackgrass. Yes, yet another herbicide resistance in blackgrass, but only a transient one. The first bit of good news that I needed was that if I spray enough Bosta onto blackgrass, it does die, if it's infected with the VOX MCS or with the VOX GFP that I showed you in the last slide. However, if I use the VOX bar virus, they survive the Bosta treatment. I often refer to this as my no statistics needed slide, and hopefully you'll agree with me that this very clear statement demonstrates it's possible to assess whether a single gene is sufficient to confer herbicide resistance to blackgrass. Now, both Bosta and GFP are gain-of-function techniques, which are incredibly useful for testing hypotheses about sufficiency, but what about necessity? One of the first genes to be used for VIGS, for virus-induced gene silencing, was phytoene desaturase, or PDS. Now, it was used back then, and it's still routinely used as a positive control because it's involved in carotenoid biosynthesis. So loss of PDS results in photobleaching of the leaves. Usually, and very usefully, white leaves are a very easy and identifiable phenotype. So I wanted to try to see if I could use this in blackgrass. I made a VIGS virus that targeted the blackgrass phytoene desaturase gene and infected blackgrass with it. And what you see here is that compared to the plants that are infected with just the multiple cloning site virus, the plants on the right have white stripy leaves. This is just what I was looking for. It's a bit hard to photograph, so I'll zoom in just on one of the leaves. And here what you see is that infection with the VIGS virus targeting the blackgrass BTS results in photobleaching of these leaves. Now, the fact that the whole leaf isn't white is once again one of the limitations to VIGS. Just like VOX, VIGS does not result in uniform silencing of the gene throughout the infected plant. However, the effect is obvious when you're looking at it. And when I grind up the whole plant and measure the amount of PDS mRNA by qPCR, what you can see is that there's a very significant reduction in the total mRNA with the VIGS infection. So excitingly, VIGS now allows us to intentionally reduce expression of a single gene in weeds. Now, this PDS phenotype is not quite as transient as it might have been. Not only does it persist for at least months after its original manifestation, it's stable when we separate out individual tillers of the plant and then replant them. The phenotype also can persist through vernalization, even if that vernalization period lasts throughout the entire period of lockdown. So plants come out of the chillers and they stay exhibiting the stripy leaves, and this continues on through flowering, as you can see on the image on the far right. In other species, this change is stable through the next generation of seedlings too. So these observations mean the range of phenotypes we can investigate and the duration of the change is much more broad for the loss of function than most transient techniques. Now, the paper didn't get picked up just because we made glowing and stripy plants, but instead because we were able to show that the VIGS used to reduce the expression of AMGSTF1, which is a glutathione S transferase that's involved in metabolic herbicide resistance, was able to reverse herbicide resistance. We did this in a sensitive biotype that dies with herbicide treatment and has a low level of AMGSTF1, and a resistant biotype known to survive herbicide treatment and have a high level of AMGSTF1. So although infection with the VIGS MCS didn't change the resistances shown here, VIGS targeting against AMGSTF1 was sufficient to make the resistant biotype look just as like the sensitive one. But before you get too excited, these VIGS and VOX vectors were designed to test hypotheses in barley and wheat, the very crops in which blackgrass grows, and the resistances in these crops is primarily achieved through detoxification of the herbicides through pathways like that use enzymes such as the GSTs, the AMGSTF1. So if we were to use these vectors out in the field, they would probably infect the crop and they would probably make it susceptible to the herbicides. So all in all, not the entirely desired outcome. But remember that these are quite big breakthroughs for weed science because we now have the capacity to do reverse genetics in blackgrass. And we can induce both gain of function and loss of function. So these are species that we've never been able to manipulate before. And excitingly, the loss of function seems amenable to making quite long-term changes that are stable through cloning and vernalization. So remember that my main aim was to bring agricultural weeds back into the molecular lab and to focus on generating genetics and genomics tools for these species. These will allow us to do hypothesis-led research in these impactful species. So although having a genome and ability to do reverse genetics may seem relatively mundane, to put this into context, although there's 262 species of weeds that have recorded levels of herbicide resistance, there's only about 17 species with published genomes or genomes that we know to be in process. And ours is the only one that I could find where we do reverse genetics in our herbicide-resistant weed species. So although these small steps will go a long way towards better understanding the genetics of weediness and moving towards identifying specific traits that we can use to substantially enhance agricultural production. So returning to poor old Alice and the Red Queen, if we bring agricultural weeds into the molecular lab, then we have the ability to quantify traits and identify the genes of weediness. With this understanding, we can inform crop breeding programs so the crops can run a little faster, or we can use it against the weeds to slow them down just enough so our weed control strategies are efficient. So I hope that during today's talk, I've convinced you that studying weeds and weediness are really important and impactful things to delve into, and that over the last couple of years, we've made some good progress at developing genetics and genomics tools for work in blackgrass, which hopefully we can then translate to other impactful species. Now, I've intentionally left a little bit of extra time, hopefully, for questions at the end of my talk, as I think I probably talked a little bit too fast. So I'll stop here and remind you that I'm Dana MacGregor. I want to thank the people involved in the plant physiology paper that came out in April, and to highlight the rest of the weeds team here at Rothamsted. I want to name those involved in the Blackgrass Genome Project and the Developing Comparative Transcriptomics Project. And of course, I'm indebted to my funders and the programs that have supported me throughout this research. I look forward to answering your questions, and thank you very much for tuning in to listen to my talk.
Excellent. Thank you very much, Dana. Indeed, it's the shortest talk we've had, I think. That's really refreshing. It's nice.
Oh, man.
So, no, it's good to give a nice introduction to the weed science and the blackgrass. Okay, so I encourage everyone to ask questions in the question box if you have anything you'd like to ask. So let me ask first of all about something nuts and boltsy. So how do you actually go about transforming a blackgrass? How did you get the genes in there?
So the transformation process is actually you create the transgenic virus, and then you propagate that in tobacco. So once you have that inoculum, then you can actually use that to infect the blackgrass. And the process by which we do that is has lots of scientific terms that I could say, but really what we do is we create small cuts on the front of the leaf, and we use our fingers to rub it in. So it's called rub inoculation, but essentially what we do is we actually directly infect the blackgrass with the VIGS or VOX virus. The VOX virus and the VIGS virus then go into the cell where they replicate, and as the plant either fights off the infection and leads to the RNA response, it leads to VIGS, or the virus replicates, it drives its translation of the heterologous gene.
And you showed it goes to the next generation. Does it go into the germline?
Well, in other species, it can definitely go through multiple generations, and actually it's gone as many as 4 generations in some other species. I don't yet have data to know whether or not it goes beyond flowering. I definitely see that the stripy leaves are manifest in flowering, but I haven't yet gotten around to being able to determine whether or not it's going on into the next generation. So stay tuned.
Okay, so right, we have a question from Gabrielle Og, and she says, very nice presentation, Dana. And she says, what strategy would you use to avoid resistance reversion transfer to the crop?
So again, I'd love to be able to take this out into the field and therefore have an opportunity to develop a resistance reversion. But at the moment, this is purely for lab-based research, because what we're doing is trying to determine ways to do the reverse genetics. Right now, we don't have any ways to do those hypothesis-led experiments in the lab, nevertheless in the field. But because it's a very specific thing, RNAi is based on 21 MERS or 23 MERS, depending on how you're inducing it. And so the idea there is that you can make a very specific aspect that you're targeting. So there is an opportunity then to make it only target the blackgrass and not the crop in which it's going. But really, this is, at the moment, if we were to take this out and use it in the fields, not only is it a GM virus that we're infecting into the wild, but also, it's something that would infect the crop and probably make the crop sensitive to the herbicides as well. So really, we're back a long way before the model organisms are, and we're trying just to do ways to say, is this gene necessary, or is that gene sufficient to confer that phenotype?
That's the challenge, right? So when you put in grants and things, everyone expects you to be at the level that the model organisms are at, even though we haven't got any of that work yet. So the questions are coming in nicely now. So first question, the next question from Fiona Cork, and she says, great talk, Dana. And she asks, has blackgrass invaded the broad bulk plot where there's been no herbicides at all? So explain what that is first of all, and then answer the question.
Yeah, so broadbulk is part of our long-term experiment, and it is an experiment that's been running since the inception of Rothamsted. So it's 177 years old this year. And yes, we've got lots of blackgrass, especially in the parts where we don't use any herbicides. So actually, some of the early photos that I showed you were from Section 8 of broadbulk. And the nice thing about this is because we've never used herbicides on these sections, we have a purely sensitive biotype. And this is what we're using for our genome, is we have something that has never, ever seen herbicides. So those evolved resistances are not happening in that, or hope to not be happening in that particular section.
Okay. All right, so we got 2 related questions from Catherine Denby and Rebecca Morell, and they say thanks for a great talk. And Rebecca says, I was wondering if you can expand on any genes, hormones, pathways that you might have that confer weediness in blackgrass.
So weediness is kind of an interesting thing, because there's lots of ideas that it's a single trait, right? So it's if you are herbicide resistant, then you're weedy. And that's definitely the case, because if you think about selection experiments, if you can survive the herbicide, then you can outcompete your neighbors regardless of what happens, because they're all dead. So herbicide resistance is one of the major traits that we think of when we think of weediness, but also resilience, seed fecundity. I mean, blackgrass germinates after the crop has gone in, and it sets its seeds before the crop comes out. So the entire life cycle competes. So if you survive that selection of the herbicide, you will dump your entire generation of seeds for the next round to go. And about 2/3 of the seeds that are left in the seed bank will germinate that next autumn. So that's a huge population of turnover, and each plant is capable of producing thousands of seeds. So as a consequence, it's really, so high fecundity is a really important thing. Timing of germination and of seed set's an important aspect. And all of these things we know are incredibly complex traits. We know that the timing of dormancy, things like that, we you know, there's lots that could be going on there. So there's lots of traits independently, but collectively, blackgrass is just really good at doing all of them. So it avoids most of the control measures, and it makes it a really super good weed.
So that also was similar to a question that Jose Gutierrez Marcos asked when he said what key trait that blackgrass has that makes it so resistant. So I mean, I think you've answered that. It's got a bunch of things going for it in its way.
Yeah, so it's highly resilient.
Yeah.
It has high fecundity, and it has bags of herbicide resistance to multiple different modes of action. So yeah, it's a very good weed if you're a researcher, not a farmer.
Yeah, absolutely. So a question from John Bannock, and he asks, why did you use VIGS? Why didn't you use CRISPR instead of VIGS? Or have you got any plans to use CRISPR in the future?
I'd love to use CRISPR. I mean, CRISPR would be much more specific. We could make those techniques work. However, if you think about it, with CRISPR, you need the Cas9 cutting enzymes, right? And you need the guide RNAs to get in there, and you need the thing to cut, right? So we need to get one or other of those things in stably. So there are some examples where they've used VOX to drive the cutting enzymes, but these are not particularly well developed or as efficient as some of the other techniques. So these are things I'd like to investigate, but really you can't do CRISPR without something to cut and something to guide. So we have to find a way to get both of those things into blackgrass before I could start to work on those techniques.
Okay, so just to finish off, I can't remember if we've discussed this before. Have you made a population of mutagenized blackgrass that you'll be able to work on for the future? I can't remember if you've done that before.
You're telling all my state secrets. Yes, no, I mean, I'm interested in both forward and reverse genetics. Mm-hmm. So yes, I'm trying to, these are techniques because it's an annual and it's an obligate outcrosser. So these are things that are not very straightforward to do, and it takes a while to get to a population. But yes, I'm working on doing establishing forward genetic screens as well as the means to do reverse genetics. The aim here is that if you can look for phenotype, then you can be driven by phenotype and not by genotype. Right now, I'm driven by genotype. If I change this gene, what happens? But by doing the forward genetics, I can actually be driven by the phenotype. It survives Y. So yes, these are populations that I'm working on developing, but they take a while. And we also desperately need a full, really well annotated genome in order to start that work. This is a 3.5 megabase genome. It's huge. So resequencing is going to be a major effort. So I need to have the right lines with the right questions and the right data before I can start down that road.
Okay. Well, just I mean, leading on from that. So you mentioned during the talk about the genome. So what's the status of the genome at the moment?
Yeah, we're really happy with our genome actually. So our annotation and the full alignment has been frozen at this point. We're now working on developing the biological stories to hang on onto it to make it really a really high impact paper. But we're at a point now where we're really happy with our annotation. We have good BUSCO scores, we have good protein predictions, we have all of the things that you would look for in order to say that this is a good, high quality genome. Our LAI scores are 22, and essentially everything over 20 is kind of as high as you can get. So we're really pleased with our genome, and the plan is to try and make the biological story interesting as well. So it's not just another genome. So with the Weed Genomes Consortium, we're going to be developing lots of genomes in the next few years. So actually, we needed something that was going to be a really high quality story to go with it. So that's what we're working on at the moment, is making sure that the story is there too.